Hello, and welcome to the first segment of two segments on metabolism. In this first lecture segment, we will cover slides 1 through 65. In this first segment, we'll cover the first 3 bullet points: ways of thinking about metabolism, metabolism in the absorptive versus post-absorptive state, and carbohydrate metabolism. And in the second segment, we'll talk about lipid metabolism and protein metabolism. What comes to mind when you think of metabolism? When I ask this question in a face-to-face class, some common comments that students offer is that metabolism has something to do with how quickly you process food, or metabolism has something to do with how much energy you have. And then there are some common questions. What does it mean to have a fast metabolism or a slow metabolism? Does metabolism have anything to do with your body type? Do thin people have a different metabolism than heavy people? And is your metabolism fixed? Is it constant? Is it something that you're born with? Or is it something that will change or you can change throughout your life? And these are all great ideas and great questions. And I hope by the time we complete the metabolism lecture material that you walk away with a much deeper, more concrete, more very real understanding of this word metabolism. So there are many different valid ways of thinking about metabolism. And we'll go through 3 different ways of thinking about metabolism because they all have something to offer about what this word means. And so a very common textbook definition of metabolism, and maybe you remember this from our discussion very early on this semester, metabolism can be thought of as the sum of all catabolic and all anabolic reactions in the body. So the sum total of all chemical reactions in the body. Here's this image again. I use this image a lot because it's elegant and it applies at many points in our physiology course. So the first time I used this image was way back in our first lecture topic, and it comes up under the characteristics that are shared by all living things. So all living things have a metabolism, and what that means is all living things use molecules. They take larger molecules and break them down into smaller molecules, and they take smaller molecules and combine them together to create larger molecules. So every living thing, meaning you, the bacteria that's in your large intestine, your houseplants, the grass outside, all of those cells have metabolism. And metabolism can be divided up into catabolic pathways and anabolic pathways. So we just finished talking about the digestive system, and a large part of digestive system activities is catabolism, taking these large molecules that are in the foods that we eat and physically chopping them up into smaller molecules. So for example, let's say you just ate a banana and that banana has a lot of starch, and starch is a polysaccharide. So you remember that you have salivary amylase, which is a carbohydrate digesting enzyme in your mouth. You also have pancreatic amylase, and you have some carbohydrate digesting enzymes that are part of your brush border. We didn't really talk too much about them. So you have several different carbohydrate digesting enzymes in your GI tract, and they all work on taking bigger carbohydrates like starch and chopping them up into smaller molecular building blocks. And those are the monosaccharides, and monosaccharides get absorbed and they end up in your blood. So now we're talking about glucose. You have glucose in your blood. And so that glucose that's in your blood is the result of a bunch of catabolic pathways, right? That glucose originated as part of a polysaccharide, and now it's a monosaccharide, and that's catabolism. Okay, so that glucose is in your blood. We get to see what happens on the other side of this equation. What does glucose anabolism look like? Or I should say carbohydrate anabolism. Well, that molecule of glucose, let's say that molecule of glucose that's traveling in your blood, let's say it enters a skeletal muscle cell. Well, skeletal muscle cells can take that molecule of glucose and incorporate it into a much larger molecule of glycogen. Glycogen is a polysaccharide. And remember, our skeletal muscle cells have all these little bells and whistles that allow them to very easily make ATP. They can stockpile most of the ingredients that are required to make ATP, including glucose, and the way they store glucose is as glycogen. So in carbohydrate anabolism, that molecule of glucose gets incorporated to be part of a very large molecule called glycogen. So one way of thinking about metabolism is it's the sum of all catabolic and all anabolic reactions in the body. A second way to think about metabolism is as the total amount of ATP that you produce and you use every day or every week or every year. ATP is not a stable molecule, so our cells make it and use it on demand. And so the amount of ATP that you make every day equals the amount of ATP that you use every day. So you can think about it as energy turnover. And this brings us to the topic of energy budgets. So you can think about metabolism as reflecting ATP production and usage, and we can divide that up in a budget. And you're probably familiar with budgets in the financial sense. So if you have a financial budget, you have an understanding of how much money you have, how much money is coming into your bank account, and then all the different ways in which you're spending that money. So a budget might be money turnover, right? Money in and money out. An energy budget is energy turnover. How much energy comes into the body and what are all the different ways in which your body spends energy? Here's a very simple visual representation of that concept. So as animals, we take in energy from the food that we eat, right? Nobody's photosynthetic. Not yet, anyway. So we obtain this energy in the food molecules that we consume. And then the outward arrows, that's a very simplistic representation of how our body spends that energy. Our body generates heat, that's a form of energy. Our muscles, as you know, generate mechanical energy. And let's take a deeper look here. This is an overly simplistic diagram, but I wanted to use it to introduce the concept of energy budgets, in versus out. So here's a financial budget where it is the pattern of money intake and money expenditure. So look at the whole pie. What does the whole pie represent? And then maybe it's a little easier to figure out what do the individual slices of the pie represent. So how large is the pie, or what does the size of the pie represent? So let's imagine that the size of this pie, the whole pie, represents your monthly income. How much money goes into your bank account every month? And then the size of each slice represents what portion of the pie you spend in all these different ways. What percentage of your monthly income do you spend on rent or tuition or entertainment or groceries, etc.? So now that we've taken a deeper look at a financial budget, let's try to construct an energy budget for your body. So there's a pie. We're going to generate a pie chart. What would the size of the pie represent? The size of the pie would represent how much energy is coming into your body, let's say in a day. And we measure food energy as calories. It's really kilocalories, but we usually throw around the word calories. And so the size of the pie is representing the number of calories that come into your body every day, your total energy intake. What would the sizes of the slices represent? Well, that would represent how we spend energy every day. And how could we name those slices? What are the different ways in which your body spends energy every day? Let's take a look. All right, so here, notice that we have 3 different pie graphs, pie charts, and for 3 different organisms. We have a pie chart for an Adelaide penguin, for a deer mouse, and for a ball python. So as a biologist, I look at this and I say, oh, a bird, a mammal, and a reptile. They're all vertebrates. Birds and mammals are endotherms, and that means that they maintain relatively warm, stable body temperatures. Any time of day, you take the body temperature of that penguin or that mouse, and it's the same temperature throughout the day pretty much, and it's warm. So warm, stable body temperature. Versus that snake, the python, its body temperature is going to fluctuate throughout the day. And it will have to behaviorally adjust its temperature. It's not maintaining a warm, stable body temperature. Okay, so let's examine the energy budgets of these three organisms. So how do we name the pieces of the pie? Well, let's look at the key to these pie graphs. In light blue, we see basal metabolism, and we'll talk a lot more about that in just a minute. Basal metabolism refers to all the ways in which the body spends energy pretty much just staying alive. We'll talk more about that in a minute. The purple represents reproduction. And just looking at these three pie charts, it looks like all of these animals are engaged in reproduction. Fortunately, I'm not. Maybe you're not either. I'm very happy about that. In orange, we see thermoregulation. So notice that there's an orange slice for the Adelaide penguin and the deer mouse, but there's no orange slice for the ball python. It's not generating it's not burning any ATP in order to stay warm. Let's put it that way, whereas the bird and the mammal are. And then in yellow, we have activity. And by activity, we mean physical activity. That's energy that their skeletal muscles are spending. And at this point in the semester, you have a very clear idea of how skeletal muscles spend ATP. And then there's the green part, and that's growth. And notice that the penguin and the deer mouse, they don't have green pieces to their pie, but the ball python does. So in birds and mammals, they don't have indeterminate growth. It means we reach our adult size and then we stay there. Whereas snakes continue to grow throughout their lifetime. So they're adding new biomass, new body mass, and that requires energy. Here's the part where I tell you it's like a sidebar. It really has nothing to do with physiology, but I love Adelaide penguins for the following reason. They were the first organism in which prostitution was discovered. So you can fast forward if you're not interested in this story. It has nothing to do with physiology. So Adelaide penguins, here's their reproductive story. When it's their breeding season, they travel to their breeding grounds, which are these islands in the southern hemisphere near Antarctica, and they reach these breeding grounds, and as soon as they get to the shore, they set about building nests. And it's a very harsh environment, right? It's cold, it's rocky, and in order to build a good nest, meaning your eggs and your chicks are likely to survive, that requires a lot of rocks in order to lift the eggs up off the ground and protect them from the water line. And so rocks are this very valuable resource. And so males and females go around collecting rocks to build their nests, and they partner. One partner is engaged in building a nest, another one is off finding food, and rocks are a resource, just like money is a resource. And so it has been discovered that, let's say there's a male and a female couple, when the male is away, the female will go over to her neighbor's nest and steal a rock. And that neighbor, if it's the male will let her, quote, get away with stealing the rock if he can have a copulation with her. So it's like the male says, yeah, I'll let you have that rock, that very precious, valuable resource, but you need to let me have sex with you. And she does. So prostitution is the exchange of resources for copulation. Anyway, okay, back to physiology. Here are energy budgets for 4 organisms. We have the human, again, our best friend, the Adelaide penguin, the deer mouse, and a different type of snake, the indigo snake. Onto this image, we've added reproduction for humans. So notice that that woman is pregnant. So she has an extra slice in her energy budget pie. Some of her ATP is going to fuel cell division, growth of the fetus. All right, so let's just look at one energy budget for a typical human who is not pregnant. The pieces of the pie in a human energy budget are basal metabolism, and that's shown in blue. Specific dynamic action, that's a very fancy sounding word. It's not very fancy at all. I'll explain it in a minute. Specific dynamic action shown in gray, and then physical activity, that's your skeletal muscle movement shown in orange. All right, so let's take a look at each piece of the pie. Basal metabolism. And we often talk about basal metabolism as a rate. So the amount of energy that you spend in a 24-hour period, right? A rate is something over time. So the amount of energy required to maintain your body at rest over a 24-hour period would be your daily basal metabolic rate. So the amount of energy required to maintain your body at rest. And when I say rest, I mean rest. Like aside from being in a coma, how restful could you possibly be? How much other energy expenditure could you possibly eliminate? In order to measure BMR, we're going to try to eliminate all extra energy expenditure that comes on top of you just staying alive. So in order to measure your BMR, and we can do it, we need to minimize, number one, the activity in your skeletal muscles. So no extra ATP expenditure through that contraction cycle. So you're laying down, you're perfectly still, not even your eyeballs are moving, your eyes are closed. Number two, we need to minimize the activity of your digestive system. There's a whole lot of secretion and movement of smooth muscle in your digestive system. Let's shut that down by having you be in the fasting state. You've already processed your last meal. There's pretty much nothing in your GI tract. Number three, thermal regulation. You need to be in what's known as your thermal neutral zone. And that means you are not spending any additional energy to maintain your body temperature. You're not trying to heat up. You're not trying to cool down. So the temperature of the room is perfect. Number four, you are not stressed. You are not thinking too hard. You're not worried. You're not even elated. You're psychologically neutral. Number five, you have a very mellow immune system because you are not actively fighting an infection. So your immune system cells are not particularly busy. They're just going about their normal daily business. And then of course, number six, you're not growing a new human. Okay, so this is what it would look like if we were going to measure your basal metabolic rate. Notice that she has a mask covering her head. This is so that we can measure the gases. We know the composition of the air that she's inhaling, and we can evaluate the composition of the air that she's exhaling. So we know how much oxygen she's consuming, and we know how much carbon dioxide she's producing. And later on today, or in this segment, we're going to look at the overall chemical equation for cellular respiration, and we understand the stoichiometry here quite well. We can calculate pretty reliably if we know how much oxygen she's consuming, we know how much ATP she's producing. And if she's producing that much ATP, she's using that much ATP. And so this lady is satisfying all of the requirements on the previous slide. She's not mentally stressed, she's in the fasting state, she's motionless, etc. And so we'll be able to calculate her basal metabolic rate based on the oxygen that she's using and or the carbon dioxide that she's producing. Okay, so back to this energy budget. For this lady, she's just a typical person. About 60% of the ATP she spends every day, she's spending it on basal metabolism, just staying alive. The rest of the energy budget, the gray piece and the orange piece, are spent above and beyond that, and we'll talk about those other two pieces. So physical activity, that's the most concrete, the most obvious one, the one that you're most familiar with because we've studied the skeletal system, the muscular system. And so even if you're sitting up, even if you're sitting still, there are muscles that are keeping your head up all day long. There are muscles that are keeping your torso erect. So even just sitting up counts as body movement. And then specific dynamic action, I make fun of this term because it really doesn't mean anything. Who decided to give it that name? They don't get the award because it's a meaningless term. It sounds all fancy pants and highfalutin, but all it means is the amount of energy that your body requires or spends in order to digest and absorb your food. So we just spent a lot of time talking about the digestive system. And it's actually quite a bit of energy. About 10% of your calories that you spend every day are spent obtaining more calories. And that's just the digestive part. Never mind the grocery shopping and the chopping and doing the dishes. Does that all count? No, that's all part of physical activity. The digestion and absorption, the calories that are spent there are called specific dynamic action. It's also known as the thermic effect of food, the heat generating effect of food. And the byproduct of every biochemical reaction is heat, right? That's the second law of thermodynamics. So your SDA goes to support the activity of the smooth muscle in your GI tract from your mouth to your anus, and then that whole variety of glands that we talked about recently. Okay. So here's our whole pie and a question. Which portions of our energy budget can we control? Which ones can you modify? Can you change the size of any of these pieces? Yes, you do have some control over how large some of the slices of the pie are. So let's look at number 1, the gray slice. On a daily basis, you can control how much energy you spend in physical activity. So this is the difference. Think about the difference between a sick day where you are in bed for pretty much the whole day. Maybe you get up to get a mug of tea or you go to the bathroom, obviously, but you really are in bed all day. Versus a very busy day where you are running around at work, you don't sit down for even a moment. And so the size of that slice is going to vary from one day to the next. Or maybe you can think about a day in which you get some good physical activity, some good exercise, versus a day where you don't exercise at all. And then number 2, the blue slice, the biggest slice, that's your BMR. It turns out we can't modify your BMR on a daily basis, but over time, if you add more muscle mass to your body, you can change your BMR. Remember, muscle tissue is very metabolically active. It has a high demand for ATP, even when you're not physically active. Muscle tissue takes a lot of energy just to maintain because it's got all those proteins in it, right? The thick and the thin filaments, the actin, the myosin, the troponin, etc. Those proteins take a lot of energy to maintain. So if you have more muscle mass, you really burn more ATP even when you're sleeping. So if you have a regular exercise routine and you have a higher percentage of muscle mass than the typical person or the couch potato, then you have increased this slice of your energy budget. Okay. We've been through 1 and 2. Number 3, another way of thinking about metabolism, and this is probably where we will spend the most time going forward from this point. We'll think about all the ways in which your body uses or stores substances. So substances, we mean molecules. We're going to talk about all the ways in which your body uses or stores carbohydrates, all the ways in which your body uses or stores lipids, all the ways in which your body uses or stores protein. And remember, those are 3 out of the 4 biological molecules. Nucleic acids, yes, you eat them, you digest them, You absorb their molecular building blocks, but they don't make up a significant portion of your caloric intake. And generally speaking, we don't think of nucleic acids as feeding into cellular respiration. You don't really use them as a fuel source, so we ignore them here. All right, so all the ways in which your body uses or stores substances. We have to think about, are these substances already present in your body, or are they incoming substances? Are they newly arriving? And so if you're in the post-absorptive state, you have finished absorbing your last meal. There are no incoming nutrients. Nothing's flooding into your blood or into your lymph across the GI tract. And your body needs to make do with what it already has. That's the post-absorptive state. But if you're absorbing your most recent meal, you are in the absorptive state. And so there's this influx of new carbs, new lipids, new amino acids, and your body gets to make decisions about how to utilize those incoming substances while you're in the absorptive state. Moving forward in this lecture, we'll talk about carbohydrate metabolism. In segment 2, we'll talk about lipid metabolism and protein metabolism. And again, in the post-absorptive state, it's been a long time since your last meal. Your GI tract is essentially empty. There are no nutrients coming into your bloodstream. So your body needs to make do, needs to maintain homeostasis with all of the molecules that it currently has on hand. And so we'll take a look at what does your body do with the carbohydrates that are already in your body? How does your body handle the lipids that are already present? How does your body handle the proteins that are already present in your body? And then in the absorptive state, that's when there's this influx of new molecules across your GI tract. And we'll talk about what your body does with carbohydrates as they're coming in, what your body does with lipids as they're coming in, and what your body does with amino acids as they're coming in. Okay, here we dive into carbohydrate metabolism. And of the 3 types of molecules, carbs, lipids, and proteins, we definitely spend the most time on carbohydrate metabolism because glucose is your cell's preferred fuel source for making ATP. So here, under the heading of carbohydrate metabolism, we will study ATP synthesis, also known as cellular respiration. But please keep in mind, tuck it away until we get there, that your cells can also use lipids and amino acids to make ATP, but the preferred fuel is carbohydrate. So here's a lot of new vocabulary. There are a lot of glucogly words under carbohydrate metabolism, but fortunately, they're good terms. You can look at the word and figure out what it means, and I'll give you those tips along the way. These words are not specific dynamic action. We're specific dynamic action. What the heck does that mean? You can't figure it out based on the term. That's not true for terms like glycogenesis, glycogenolysis, and gluconeogenesis. We'll get there. All right, so carbohydrate metabolism. That's sort of an umbrella that covers all the ways in which carbohydrates are used, stored, manufactured, and regulated. And here's a list of topics that we'll cover as part of carbohydrate metabolism. All right, don't get scared. When I was an undergraduate, I probably had to memorize various pathways in cellular respiration several times. And I know how intimidating it can be, and I am certainly not going to ask you to memorize any biochemical pathways. I would like you to know an overview, the steps that are involved in using glucose as a fuel for creating new molecules of ATP. And that's what we're looking at here. There are 4 steps in cellular respiration where glucose is the starting fuel source. So the first step is glycolysis, and glycolysis manufactures or results in the ingredients for the next step. The next step is the formation of acetyl coenzyme A, and acetyl coenzyme A is the ingredient for the third step, the Krebs cycle. And the Krebs cycle generates the ingredients for the fourth step, the electron transport chain. So there's a clear flow here. 1 into 2 into 3 into 4. So what this word means, cellular respiration. It's the stepwise catabolism of a fuel molecule. Let's just stop there for a moment. So step by step, a fuel molecule like glucose is broken apart gradually, little bits at a time, in a very controlled fashion. The stepwise breakdown of a fuel molecule by which the energy extracted from the fuel molecule is transferred into new molecules of ATP. So you remember the law of conservation of energy, right? That was also a very early lecture this semester and probably something that you were initially taught in 3rd grade. It means that energy isn't created or destroyed, it just moves around. It gets transferred from one form to another. So there's energy in glucose molecules, and that energy gets extracted during cellular respiration and put into the chemical bonds in a molecule of ATP. That's cellular respiration. And here we come to the second law of thermodynamics, which just says that energy transfer is inefficient. So anytime you convert energy from one form to another, some of that original energy is necessarily lost as heat. And heat is not a particularly useful form of energy, right? Heat can't fuel your muscle contraction. Heat doesn't run your sodium-potassium pumps. Heat can keep your body warm, and that's important. So we are back to this picture. I use this picture so much, where instead of food molecules, I'd like you to consider them fuel molecules, right? What does it mean to add fuel to a fire? It means throw another log on, where the molecules that are in the wood have energy that keep the fire burning. So the molecules in the food that you eat are legitimately fuel molecules because they power ATP synthesis. Okay, here are those 4 steps in cellular respiration, and we are specifically talking about aerobic cellular respiration, which I'll talk more about in just a moment. Step 1 is glycolysis. Step 2 is the formation of acetyl coenzyme A. Step 3 is the Krebs cycle, which is also known as the citric acid cycle. Step 4 is the electron transport chain. And again, each step creates the ingredients for the next step. So without glycolysis, we don't have formation of acetyl-CoA. Without acetyl-CoA, we don't have the Krebs cycle. Without the Krebs cycle, guess what? No electron transport chain and you die. Okay, big scary picture, not really. I will show you cellular respiration using many different visual aids because it is helpful to have a diversity of visual tools for learning the same concept. So what are we seeing here? That kind of tan pink background, that is the cytosol. And so in the cytosol of all of your body cells, glycolysis happens. And you see there on the left, there's that blue rectangle that shows glycolysis. During glycolysis, a molecule of glucose, and you remember that glucose is C6H12O6. So it's this 6-carbon molecule. It gets split apart to form 2 molecules of pyruvate, also known as pyruvic acid. And then that pyruvate moves into the mitochondrion, so sort of that tan blob in the upper 2/3 of the diagram. That's a single mitochondrion. You remember that's the powerhouse of the cell. That's where most of your ATP is produced, is inside mitochondria. So that pyruvate enters the mitochondria and it gets converted into acetyl-CoA. And that acetyl-CoA feeds into the Krebs cycle, which is also known as the citric acid cycle. So this is happening now inside the mitochondria. And the Krebs cycle, or the citric acid cycle, generates ingredients that are required to fuel what's known as the electron transport chain. So we're all the way on the right now in sort of that purplish rectangle. And the electron transport chain also is inside the mitochondrion, and this is really where there's a lot of ATP produced. So in these 4 steps, starting with a molecule of glucose and ending up with 32 to 34 molecules of ATP, this is all about energy transfer. The energy that was in the chemical bonds between the atoms in a molecule of glucose, that chemical energy gets transferred out of glucose and into ATP. It's just a very kind of long, sophisticated process that we, the community of science, we understand cellular respiration quite well. It's really cool. So that we understand the chemistry so well that we can measure your respiratory gases. How much oxygen are you consuming? How much carbohydrate are you producing? And we know pretty closely how much ATP you're producing. All right, so there are these 4 steps. Does every step manufacture ATP, or do only some of the steps manufacture ATP? Let's use this diagram to take a look. So ATP is represented sort of in that dark orange oval star shape. So let's look. Glucose manufactures a net gain of 2 ATP. So just splitting apart that molecule of glucose to make 2 molecules of pyruvic acid, that gives us 2 new molecules of ATP. The next step, when we go from pyruvic acid to acetyl coenzyme A, oh look, no ATP is formed there. No ATP is formed there, and we'll talk about what NADH is in a little bit. No ATP is formed there, but let's go to the 3rd step, the Krebs cycle. Do we get any ATP out of the Krebs cycle? We sure do. We get another 2 molecules of ATP. So we've taken the first 3 steps to break down that molecule of glucose. We've done glycolysis, we formed acetyl-CoA, we've gone through the Krebs cycle, and so far we got 2 ATP from glycolysis, we get 2 ATP from the Krebs cycle. That's a total of 4 ATP. It's not a lot of ATP. So let's take a look at the electron transport chain. Bam, that's our cash cow. We get a lot of ATP through the electron transport chain. Mitochondria are very effective at extracting energy and using that energy to generate new molecules of ATP. Oh, the Krebs cycle. Again, as an undergraduate, I think I actually had to memorize the Krebs cycle, memorize it, all the steps, all the enzymes. I think I had to memorize that 4 times. And if you have to memorize something 4 times, it means you didn't really learn it the first 3 times. So I'm not going to subject you to that. I would, however, really like you to have a strong foundation in cellular respiration, and that includes the Krebs cycle. So we see our starting ingredient is pyruvic acid at the top, and that is a 3-carbon molecule, right? During glycolysis, that 6-carbon molecule of glucose gets split, glycolysis, splitting glucose into 2 molecules of pyruvate. And that here we're just looking at 1 molecule of pyruvate, also known as pyruvic acid. And that undergoes a very short biochemical pathway. It's a very simple step. It gets converted into acetyl coenzyme A. And just in that 1 step, we can see that carbon was removed, right? We form carbon dioxide. So now we've lost a carbon atom. What happens to that carbon dioxide? Where does it go in your body? Yeah, that's a waste product of metabolism. So that carbon dioxide is going to ultimately diffuse out of the cell into your blood, and it's part of the carbon dioxide that you exhale. So this acetyl coenzyme A enters the Krebs cycle, and during the Krebs cycle, this acetyl coenzyme A is further broken down. Carbon atoms are removed, and so you can see other carbon atoms are created. Other carbon atoms are not created during the Krebs cycle. Other molecules of carbon dioxide are created during the Krebs cycle as they are removed ultimately from this acetyl coenzyme A, and those carbon dioxide molecules are also waste products that get exhaled. Another key item here, and this is new for us today, another key item here are the electron carriers. The electron carriers. So we have 2 different types of electron carriers. We have NADH and we have FADH2. And so these are little shuttles. They can pick up electrons here in the Krebs cycle, and they will move through the mitochondrion, and they will donate these electrons to the electron transport chain. So as this molecule of acetyl coenzyme A is broken down, the carbon dioxide molecules form, and these are the waste products. They have no known value in our body, and they are eliminated when we exhale. And then valuable products of the Krebs cycle include NADH. It's accepted an electron, and that electron is headed for the electron transport chain. And FADH2 has also accepted an electron. And it's okay to think about an electron as like a little packet of energy at this point. It's not really, but then it gets a little complicated. But when these electron carriers accept electrons, they're in a higher energy state, and they're going to go and donate those electrons to the next step, the ETC, the electron transport chain. All right, so NADH and FADH2 are electron carriers. These electrons are liberated during glycolysis, during the formation of acetyl-CoA, and during the Krebs cycle. And so this is part of how the energy is transferred out of a molecule of glucose and into new molecules of ATP. Here we take a look at what happens inside the mitochondrion, specifically the electron transport chain. So let's start by seeing where we're at. Here's a mitochondrion. A mitochondrion is a membrane enclosed organelle. Specifically, it's a double membrane organelle, meaning it's got two membranes. There's a membrane that surrounds the exterior of the organelle, and that's called the outer membrane. And then there's a membrane that forms these cristae, these folds, and these little mini compartments inside the organelle. And so that's the inner membrane versus the outer membrane. And what we're looking at in this picture here, this membrane here, and you remember membranes are phospholipid bilayers with proteins stuck in them. The same holds true for both membranes of the mitochondria. And so we are looking at a tiny section of the inner membrane inside a mitochondrion, where it's a double layer of phospholipids with proteins embedded in it. And we'll talk about those proteins in just a moment. But this inner membrane creates subcompartments within the mitochondria. And so in pink, we have the mitochondrial matrix, right? And then on the other side of the membrane, we have the intermembrane space. So in blue, we have the space, the region of the mitochondrion that exists between the outer membrane, which isn't really shown here, but the outer membrane of the mitochondrion would be here. And then the inner membrane, which is labeled, right? It's right here. So we have these two subcompartments within the mitochondria, the mitochondrial matrix and the intermembrane space, and they are separated by a phospholipid bilayer with proteins embedded in it. So the proteins that make up the components of the electron transport chain include 4 protein complexes, and they're labeled down here, complex 1, 2, 3, and 4. And these protein complexes can accept an electron and donate an electron to the next complex in the chain. So let me change colors and let's check out our friends, the electron carriers that we talked about previously. So one of our electron carriers is NADH and the other one is FADH2. And these molecules have already picked up an electron. They either picked up that electron that was liberated during glycolysis, or maybe that electron came out of the formation of acetyl-CoA, or maybe that electron was liberated during the Krebs cycle. It doesn't matter. NADH and FADH2, they both have an electron and they donate it to one of the complexes that makes up the electron transport chain. And when they donate their electron, they go from a high energy state back to their low energy state. So NADH will revert to NAD, And NAD, you know what it'll do? It'll go back and pick up another electron. So NAD will just cycle throughout this high energy state. When it has an electron, this NADH, it'll donate the electron and become NAD again, and it'll go back and pick up another electron. So they're electron shuttles, they're electron carriers, and the same thing is true for FADH2. It'll donate its electron, it'll go back into its lower energy state, it'll go get another electron, it become FADH2, donate that, etc. So let's take a look at these protein complexes. Let's look at complex 1, we'll look at complex 3, and we'll look at complex 4, because they all share an important characteristic. All of these complexes, 1, 3, and 4, they are hydrogen ion pumps, also known as proton pumps. So they use the energy in that moving electron. They use that energy to drive a hydrogen ion uphill against its concentration gradient. This is a form of active transport. They use that energy, not in ATP, but the energy in a moving electron to drive hydrogen out of the mitochondrial matrix and into the inner membrane space. So let's take a look at each of these complexes. There is a whole bunch of hydrogen ions and a dark black arrow indicating that as this electron moves from NADH through complex 1 and over to this intermediate called coenzyme Q, as that electron moves, the energy is used to drive one hydrogen ion out of the mitochondrial matrix and into the inner membrane space. And that electron continues to move from complex to complex. Complex 3 also pumps hydrogen ions. Complex 4 also pumps hydrogen ions. And there are a couple of intermediaries here that don't pump anything. This is coenzyme Q and this is cytochrome C. Important necessary ingredients for the electron transport chain. So the key outcome of the electron transport chain is we get this very steep hydrogen ion gradient where there is lots of hydrogen in the inner membrane space and there's very little hydrogen in the mitochondrial matrix, and it's a gradient, and gradients represent a form of potential energy. So now we have a lot of stored energy because this membrane, this phospholipid bilayer, is preventing these hydrogen ions from flowing downhill, from undoing the gradient. And so it's like that dam holding back the water. The water wants to flow, but it can't. It's being held back by a dam. Well, these hydrogen ions want to flow, but they can't. They're being held apart. They're being held back by this phospholipid bilayer. So the major point of the electron transport chain is to generate this very steep hydrogen ion gradient. So let me ask you, which compartment has the higher pH? If we could test the pH of the fluid that's in the inner membrane space, and we test the pH of the fluid that's inside the mitochondrial matrix, that compartment, which one has the higher pH? The fluid that's in the mitochondrial matrix, because it has a very, very low concentration of hydrogen ions. So it is very basic. And the fluid that's in the fluid that's in the inner membrane space, that has a super high concentration of hydrogen ions. And so it's very acidic. Okay, so what do the mitochondria do with this enormous hydrogen ion gradient? What's the purpose? The purpose, again, is to make ATP. And so we get to look at one of the coolest molecules in living things, and that's on the next slide, and it's called ATP synthase. Great name. All right, so ATP synthase, right? You don't really have to wonder too long. Well, what does that molecule do? Gosh, I don't know. Oh, could it synthesize ATP? Yes, it does. So it's an aptly named molecule. It would get the award for a great name from me. And let's zoom out for a minute and take in the bigger picture here. So we are looking at a tiny region of a mitochondrion, And let me use this diagram while we have it up to reinforce that mitochondria have two different membranes in sort of this gray pink color. This is the outer membrane. And in orange, this is the inner membrane. And this is where we find the proteins that make up the electron transport chain. And that gives rise to two different compartments in the mitochondrion. In sort of this purplish gray color, this is the mitochondrial matrix, and which has a very low concentration of hydrogen ions, and it's hard for me to draw, but you can see the light blue region here. This is the inner membrane space. The space that's between the outer membrane and the inner membrane is called the inner membrane space. And that's where we find that high concentration of hydrogen ions. And then notice that the artist drew this little square here to say, hey, I'm taking this little part of the picture and I'm blowing it up here so that you can see what's happening in that tiny little square. So here we see the inner membrane of the mitochondrion, and we see the artist just chose to represent the entire electron transport chain in this one sort of reddish brown blob, right? But the electron transport chain uses the energy of bouncing electrons, moving electrons, to pump hydrogen ions out of the matrix and into the inner membrane space. So we have this very steep hydrogen ion gradient. And so what does ATP synthase do? ATP synthase is a channel, or part of it is a channel. It's a channel that allows hydrogen ions to flow along their electrochemical gradient. And as these hydrogen ions flow through this protein, through this ATP synthase, this protein is going to change shape in such a way, it's very similar to a turbine in a hydroelectric power plant. It's amazing. This protein will change shape. It undergoes a conformational change in a way that allows it to catalyze this reaction. So ATP synthase is an enzyme, right? It ends in ACE. It's an enzyme. It's also a channel. And as an enzyme, it catalyzes this reaction right here. It takes ADP, adenosine diphosphate, and tacks on the third phosphate to create adenosine triphosphate. So the energy that exists in the concentration gradient for hydrogen ions is used by ATP synthase to generate new molecules of ATP. So we can put all those parts together. We're zoomed out. We see the different complexes that form the electron transport chain. Let me remind you that here is a very low concentration of hydrogen ions. Here's a very high concentration of hydrogen ions. We see our best friends, the electron carriers, NADH. I don't see FADH2 represented here, but that's okay. And so the electron carriers donate their electrons. Here's our bouncing electron. It'll bounce from one protein to the next, to the next. Some of these proteins are also hydrogen ion pumps, right? So we see this accumulating hydrogen out here in the inner membrane space. And then here we have our friend ATP synthase that is constantly generating new molecules of ATP using the energy in that existing electrochemical gradient. All right, the next thing that we get to add on here is, why do your cells need oxygen? It's a fundamental question. We could have asked that question on day 1 of the semester. Why do you need oxygen to live? Most people don't know. Here's why you need oxygen to live. Oxygen is what's known as the terminal electron acceptor. So it plays a role in the electron transport chain. That bouncing electron has to end up somewhere. It's got to go away. How do we get rid of that electron after we've used it, so to speak? And you know what? That electron couples with a molecule of oxygen to form new molecules of water. So a byproduct of the electron transport chain is the formation of new water molecules. And this is what happens to the oxygen that you consume, right? That oxygen, we've studied the respiratory system. You know that oxygen diffuses out of your alveoli into your blood, out of your blood at the capillaries into your cells, and then here's what your cells do with it. That oxygen diffuses into the mitochondria. That oxygen accepts electrons as the end of the line in the electron transport chain. That oxygen undergoes a chemical reaction with these electrons, and it becomes a brand new molecule of water. And so you're manufacturing new molecules of water inside your mitochondria all the time. It's called metabolic water, and it forms a tiny little part of your water balance. All right, so that's why you need oxygen. Another less important but fun fact, are you familiar with hydrogen cyanide? It's a poison. It will kill you. It's popular in movies and spy novels. So how does cyanide work? Cyanide binds to this protein in the electron transport chain. It's called cytochrome C. And when cyanide binds to cytochrome C, it blocks the flow of electrons. So the electron will start to bounce from one complex to the next. It'll hit cytochrome C and it'll stop. It won't continue to bounce. And so cyanide disrupts the electron transport chain. So it disrupts your mitochondria's ability to generate ATP, which is why it results in death. All right, let's put all those pieces together. It's a big, long story, I know. So we start at the very beginning, top left of this diagram, the purple rectangle. We start with a single molecule of glucose, and that's a 6-carbon molecule, C6H12O6. Remember, we have to account for all the energy. We have to account for all the matter. We can trace the fate of every single one of those atoms in that initial molecule of glucose. They all go somewhere. So we perform glycolysis. We take that one 6-carbon atom and we make 2 3-carbon atoms of pyruvate. We generate a little bit of ATP. We donate electrons to an electron carrier. Next step, we take that pyruvate and we form acetyl coenzyme A. And in this step, we generate our first molecules of carbon dioxide and we donate electrons to more electron carriers. So we sort of we power up another couple molecules of NADH2. And then we take the next step, and that acetyl coenzyme A goes through the Krebs cycle where we generate some more molecules of carbon dioxide, this waste product. We generate a couple new molecules of ATP, and probably most importantly, we power up many more electron carriers. So we have powered up electron carriers in glycolysis, in the formation of acetyl-CoA. We have powered up electron carriers in the Krebs cycle. And notice that all of these electron carriers donate their electrons to the electron transport chain. And I love this. Follow the bouncing electron. And we can follow the bouncing electron to the terminal electron acceptor. The end of the line is a molecule of oxygen. And this allows the electron transport chain to keep flowing, right? Those electrons have to get off somewhere to make room for the next electrons. And where they get off is oxygen. We manufacture some water. And of course, this creates a very steep hydrogen ion gradient that allows ATP synthase to do its magic. Only it's not magic, it's science. Whew, are you tired yet? I am. I'm exhausted from this journey. But let's take a step back and be reminded about these blood gases. We talked a lot about them when we covered the respiratory system and the cardiovascular system. So let's take a look at carbon dioxide. Now you have an understanding of where, why, how carbon dioxide is produced. The carbon in carbon dioxide came from a fuel molecule. We've been looking at glucose. Later on, we'll talk about lipids and amino acids. But that carbon dioxide that you're exhaling, that carbon atom, came from a molecule of glucose. And it was produced inside your cells, either in the cytosol or in the mitochondrion, depending where that carbon dioxide was formed. And that carbon dioxide diffuses out of your cells and into your blood. And you remember the different ways that carbon travels as it travels in your blood. Some of that carbon is just straight up dissolved carbon dioxide. Some of that carbon, let's see, is it represented on this slide? It sure is. It's carbaminohemoglobin. Hard to say that without smiling. It's still true. And then most of that carbon dioxide travels in the form of bicarbonate ion here. All right, and then what happens during carbon dioxide unloading? You know it. That dissolved carbon dioxide, well, that's still CO2. It's very easy. It just diffuses out of your blood and into an alveolus, and then it gets exhaled. That carbaminohemoglobin, check it out. The carbon dioxide dissociates from hemoglobin, and it too is now dissolved carbon dioxide, diffuses into the alveolus, and you remember that bicarbonate buffer system where this carbonic acid is going to combine this bicarbonate ion is going to combine with a hydrogen ion to form carbonic acid, and that will dissociate into water and carbon dioxide. This dissolved carbon dioxide will make its way into the alveolus. Question: why is oxygen necessary? What role does it play? So imagine your very bright and curious 12-year-old cousin asks you, hey, why do people die without oxygen? Can you answer her? I hope you can. This is what's known as a summary equation. If we take all of the steps in cellular respiration, so that means glycolysis, forming acetyl-CoA, the Krebs cycle, the electron transport chain, if we create a summary of the molecular interactions, we come up with this equation where we know that if we start with just one molecule of glucose and we form 36 molecules of ATP, in order to do that, we also consume 6 molecules of oxygen, so 6 molecules of oxygen per 1 molecule of sugar, glucose, and then our products are 6 molecules of carbon dioxide. We form 6 molecules of water in the process of using glucose as a fuel source to make ATP. And remember, the law of conservation of mass and the law of conservation of energy apply. So the amount of energy on this side of the equation equals the amount of energy on this side of the equation. The number of atoms of each kind on this side of the equation equals the number of atoms of each kind on this side of the equation. We understand this process so well that we can measure how much oxygen you consume or how much carbon dioxide you produce, and we can tell how much ATP you make. So that is cellular respiration using glucose as the fuel source. We can also use amino acids and fatty acids as fuel source. We can break apart those chemical bonds and liberate that energy to power up those electron carriers to make ATP. And we'll talk about that a little bit later. But we have more to talk about that comes under that umbrella of carbohydrate metabolism. This is a good time to take a break. Cellular respiration is involved, it's dense, it's exciting. We're going to shift gears a little bit to talk about carbohydrate anabolism, and it would be a good time for a palate cleanser, so to speak. So maybe pause, maybe keep going. I'm going to keep going. Okay, so when there is more glucose in your body than your body currently needs to support ATP synthesis, you've got all the glucose you need to make as much ATP as you need in the moment, that excess glucose is stored. And it's stored a couple of ways. The easiest way to store glucose is to use it to build new molecules of glycogen. And I'll remind you that glycogen is a polysaccharide, and there's a picture of glycogen here where each one of these subunits is a molecule of glucose. So a molecule of glycogen is huge. It can contain thousands of individual subunits of glucose. And so the process by which we take individual units of glucose, individual molecules of glucose, The process by which we combine those glucose molecules together to make glycogen, that's called glycogenesis. It's a good word. Glyco, like because we're making glycogen, and genesis, to create. So to create new molecules of glycogen is called glycogenesis. And two types of cells in our body can do this. Skeletal muscle cells, which we've talked about previously. Skeletal muscle cells can store glucose in the form of glycogen. So they have their own on-hand fuel supply in order to support ATP synthesis. Your liver cells can also store glucose as glycogen. The opposite of glycogenesis is glycogenolysis. So glycogenolysis is the catabolism of glycogen, the splitting apart of the polysaccharide to form monosaccharides. And this is a good word too, glycogenolysis. Lysis means splitting. So the splitting of glycogen is called glycogenolysis. And when this happens in your liver cells, it's because your blood sugar is running low, you haven't eaten in a while, and we need to maintain the concentration of glucose in your blood, or you die. And so to do this, your liver cells will break down glycogen and release that resulting glucose into your blood. In class, I usually ask how many of you haven't eaten today? And there's, no matter the time of day, always a couple of hands go up. Even if it's an evening class, a few hands go up. Yep, I haven't eaten all day. And those people are running all of their blood glucose has come from their liver at that point because they've been in the post-absorptive state for probably since yesterday, right? They went through the whole night, they've gone through the whole day where there's no incoming nutrients coming through their GI tract. So what's maintaining their blood sugar? Their liver. Their liver is producing glucose through glycogenolysis. Glycogenolysis also happens in our skeletal muscle in order to support ATP synthesis. All right, so I just mentioned this. Your liver cells, most of your liver cells are hepatocytes, and they wear 8 million hats. They do so many different things. Your liver cells are very busy, very capable, very well-rounded cells. One of the many functions they have is to generate new molecules of glucose through glycogenolysis in order to maintain your blood sugar. Your skeletal muscle cells also break down granules of glycogen in order to produce glucose so that they can make new molecules of ATP to support muscle contraction. All right, our next glue-gly word is gluconeogenesis. Gluconeogenesis is the formation of brand new molecules of glucose using non-carbohydrate starting ingredients. Let's look at the word. Gluco, we're going to generate new molecules of glucose. Neo means new. And then genesis is the creation of. Gluconeogenesis, the synthesis of glucose from non-carbohydrate sources. So glucose in your blood is so important to your life that natural selection has endowed you with the ability to create glucose from other types of molecules. You can imagine that there would be a strong selective pressure. If you know that without glucose you die, that's a pretty strong selective pressure in favor of organisms that have this capability of creating new molecules of glucose out of other stuff, where that other stuff includes amino acids. Certain amino acids, your liver cells can take them and magically, and when I say magic, I mean science, and magically convert them into new molecules of glucose. Glycerol, which maybe you remember, is a building block of triglycerides, right? A triglyceride is 3 fatty acids attached to a unit of glycerol. And then lactic acid. Lactic acid can also be converted into new molecules of glucose. And maybe you remember lactic acid from our conversation on skeletal muscle metabolism, right? Following glycolysis, if there's not enough oxygen being supplied to the muscle cells, that pyruvic acid will not enter the mitochondrion, but rather it will be converted into lactic acid. And that lactic acid diffuses out of your muscle cells, back into your blood. And you know what? Your liver can turn that lactic acid back into glucose. So the whole purpose of gluconeogenesis is to guarantee, to ensure glucose availability even when carbs are scarce. So under what circumstances would carbs be scarce? Where would carbs, like what's going on with you and your body if carbohydrates are hard for your cells to find? Maybe you're doing a low carb diet. Your diet consists mostly of proteins and fats. Well, you need glucose to live. This is how you maintain your blood glucose levels, gluconeogenesis. Another circumstance under which carbohydrates are scarce is a low calorie diet or starvation. So why do you need glucose to live? I have mentioned previously that other types of molecules can be used for cellular respiration. Yep, you can use amino acids to make ATP. You can use fatty acids to make ATP. Why do you need glucose at all? Well, there are a couple of cell types that rely on glucose. They're not very good at using the alternate fuel sources. So let's start with the red blood cells. Remember, red blood cells don't have any organelles. They don't even have a nucleus. So they don't have any mitochondria. So they can't do aerobic cellular respiration. They have no mitochondria. How are they going to make ATP? They rely solely on glycolysis, and that happens in the cytosol. And then neurons. Neurons in the brain and the spinal cord, for example, well, not for example, but those neurons that are part of your central nervous system, they exist inside what's known as the blood-brain barrier. So they don't have great access to the other fuel molecules. They really do rely on glucose because amino acids and fatty acids don't easily penetrate that blood-brain barrier. These neurons, remember, they're pretty delicate, they're picky, they have a whole host of servants called glial cells that supply them with nutrients. Neurons really rely on glucose to make ATP. Okay, so imagine a body that's in the post-absorptive state. These are the people in class. Raise your hand if you haven't eaten today. That's who we're talking about here. What's maintaining their blood glucose? These two processes, glycogenolysis. They have stored glucose as glycogen, and that glycogen is being catabolized. It's being doled out. Molecule of glucose by molecule of glucose, it's being released into their blood. Simultaneously, their liver cells and their kidney cells are creating new molecules of glucose using non-carbohydrate starting ingredients. So they're taking amino acids and creating glucose and adding that to the blood. So let's look at the red questions on this slide. Which arrows indicate glycogenolysis? Can we figure that out? Sure, we can. Here's glycogen. This black arrow right here indicates glycogenolysis, breaking apart that polysaccharide into individual units of glucose. And then the second question is, which arrows indicate gluconeogenesis? So we start with something that's not a carbohydrate and we end up with glucose. What's going on here? Oh, all of these. This is other substrates. So all of these are non-carbohydrate sources, amino acids, glycerol. I'll just say that these other substrates are maybe lactic acid, but none of these are carbohydrates in the strict sense, and they can all be used to produce new molecules of glucose, and that's gluconeogenesis. And notice that all this glucose is glucose. It doesn't matter where it came from, how it was produced. It's all C6H12O6, and it ends up in the blood, and you stay alive. Here's another way to visualize what we've been talking about, that there are two ways in which liver cells can add glucose to the blood. We have glycogenolysis here, and we have gluconeogenesis here. The end result is glucose, 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 and that glucose gets added to your blood. The next addition here, we see this word glucagon, as if you need another glue-gly word in your life. You do. Glucagon is a hormone that we'll talk about as part of blood glucose homeostasis. So the concentration of glucose in your blood fluctuates throughout the day. So it is a physiological variable. It is a controlled condition. It does change throughout the day. In the fasting state or in the post-absorptive state, a normal blood glucose level is between 70 and 110 milligrams per deciliter, where a deciliter is 100 milliliters. Here's what blood glucose for a typical person might look like throughout the day. So they're asleep and their blood glucose is roughly 90 milligrams per deciliter. They wake up, they eat breakfast. Now they're in the absorptive state. So that glucose is coming in from the carbohydrate that was in their breakfast. And as part of blood glucose homeostasis, that we'll talk about in a little bit, that blood glucose comes back down to around 90 again, and then they eat lunch, and clearly there were more carbohydrates in their lunch, and then there were in their breakfast, or maybe the meal was larger, their blood glucose goes up, and then it comes back down to roughly 90 mg per deciliter, and then they eat dinner. This person, let's say they had macaroni and cheese and mashed potatoes and garlic bread, who knows, they ate all the carbs, and then maybe they had a, I don't know, the piece of pie for dessert. They had all the carbs, this large increase in the concentration of glucose in their blood, and then as part of blood glucose homeostasis, we go back to the baseline. So we're going to talk about what controls the concentration of glucose in your blood as part of blood glucose homeostasis. All right, so we have the same axes here. We have blood glucose concentration on the vertical axis and time on the horizontal axis. And we're looking at how blood glucose concentration changes according to the type of meal that you've eaten. So let's say somebody just ate the mashed potatoes and the macaroni and cheese, and then they had the piece of cherry pie. This would be the carbohydrate-rich meal, which is full of carbs, and those carbs are catabolized in your GI tract and absorbed as glucose. And so we get this pretty rapid rise in blood glucose. But now let's say that somebody had the low carbohydrate meal. Maybe they ate an omelet with cheese, right? That's a lot of protein, it's a lot of fat, it doesn't contain a lot of carbs. So there's not a lot of glucose coming in across their GI tract. There's some, right? Eggs have some carbohydrate, cheese has some carbohydrate, but not a lot. So we get this much milder increase in blood glucose following this low carb meal. So we can think about the concentration of glucose in the blood, and we can use the source sink framework. Let me step back and give you another example of where we can use the source sink framework. We can think about how much carbon dioxide is in the atmosphere. So we're not talking about physiology for the moment. We are thinking about climate change. And you know that Earth's overall average temperature is related to the concentration of carbon dioxide in the atmosphere. And in trying to understand that, we can think about sources of carbon dioxide and sinks for carbon dioxide. Sources are things that add carbon dioxide to the atmosphere. Sinks are things that remove carbon dioxide from the atmosphere. So sources of carbon dioxide, you know this, burning fossil fuels adds carbon dioxide to the atmosphere. You exhaling adds carbon dioxide to the atmosphere. So those are sources. And then sinks. Trees, plants growing, they capture carbon dioxide out of the atmosphere. They use that carbon dioxide to manufacture sugar, and then they take that sugar and they manufacture other biological molecules like polysaccharides, like cellulose to form their cell walls. And so much of the body of a plant, like a redwood tree, there's so much carbon there, and all of that carbon originated as carbon dioxide in the atmosphere. So growing plants are carbon sinks. So let's use that model to think about sources and sinks of glucose in your blood. So a source is anything that adds glucose to your blood, and a sink is anything that takes glucose out of your blood. So sources, how does glucose get into your blood? Two ways. Absorption in your GI tract. The carbohydrates that you eat, they end up as glucose in your blood. Number two, from your liver. So this happens when you're fasting. Your liver adds glucose to your blood as a way of keeping you alive. And those two processes are glycogenolysis and gluconeogenesis that we talked about previously. Sinks. How does glucose come out of your blood? Where does it go? It goes into your body cells to fuel cellular respiration. So all of your body cells have glucose transporter molecules in their plasma membrane. And these transporter molecules are called GLUT. There's many different variations on them, so there are subtypes. But in general, glucose transporter molecules are called GLUT. And where's my, there we go, we'll do it in green. So those yellow transporter molecules, they're transmembrane proteins, and they perform facilitated diffusion. They move glucose molecules along their concentration gradient from an area of high concentration to an area of low concentration. So this is a type of passive transport. And because these GLUT transporter molecules are in the plasma membrane, these cells are able to take in glucose. If these GLUT molecules were not in the plasma membrane, there would be no way for glucose to get into the cell. All right, so it turns out that insulin plays a role in the presence. How many of these glucose transporter molecules, how many GLUT transporters are present in the plasma membrane? That's influenced by a signaling molecule called insulin. Insulin is a signaling molecule secreted by your pancreas. It's a hormone, and it binds to its receptor. So here we see the signaling molecule binding to its receptor. And remember, receptors are proteins, and this protein is part of the plasma membrane. Insulin binds to its receptor, and it initiates a signal transduction pathway. That's a term from the past. It's still important. This is going back to cell signaling. So cells in the pancreas have released a signaling molecule that's now sending the message to, maybe this is a muscle cell. So this muscle cell is responding to insulin by inserting these molecules of GLUT into the plasma membrane. So this is a mechanism right here. Let me change colors. Let's go to red. This mechanism right here is very similar to something we talked about in the urinary system. Do you remember ADH, that hormone? And do you remember aquaporins, those membrane proteins? Well, just like aquaporins, GLUT hangs out in a storage vesicle. And when the cell receives the signal, in this case, the signal is insulin, when the cell receives the signal, this storage vesicle fuses with the plasma membrane to insert these proteins into the membrane. Very similar to aquaporins, except these are molecules of GLUT, and the signaling molecule is insulin. And this happens in most of your body cells, have receptors for insulin. And so under the influence of insulin, your body cells become more permeable to glucose. If there's a concentration gradient for glucose, and there is, that glucose will now be able to easily enter your body cells. And so insulin is secreted, your body cells become very permeable to glucose. Glucose enters your cells, it leaves your blood, and that causes your blood glucose to decline. So insulin is a hormone that's produced by specific cells in your pancreas. And insulin, the major effect of insulin, it has other effects as well. The major effect of insulin is to lower your blood glucose by causing your body cells to be more permeable to glucose, so they take up more glucose out of your blood. A second effect of insulin is that it causes your liver cells and your skeletal muscle cells to take that glucose and to store it as glycogen. So let's take a quick look at the pancreas. We've talked about the pancreas before because we recently covered the digestive system. And the pancreas is part of your digestive system. It's also part of your endocrine system. And as part of the digestive system, we talked about these cells. These are the acini cells, or sometimes they're called acinar cells. These cells here manufacture the components of pancreatic juice. So they manufacture pancreatic amylase, they manufacture trypsinogen, and those are components of pancreatic juice that travel through the ducts and ultimately the ducts into the duodenum, right? That's part of the digestive system. Here, when we're talking about blood glucose homeostasis, we're not really talking about the digestive system at all at the moment. We are talking about the endocrine system. And so different cells in the pancreas that are part of the islets of Langerhans, or the pancreatic islets, which is this patch of cells here, these cells do not manufacture digestive enzymes. Rather, they manufacture hormones, and these beta cells, the purple guys, these beta cells, there's a beta cell, a big beta cell here. These beta cells manufacture insulin, and insulin is a very small protein hormone. And so insulin leaves these beta cells and gets into your blood, and it binds to insulin receptors on the plasma membrane of pretty much all your body cells. And we can see the example here is a skeletal muscle cell. All right, so let's look at the two halves of blood glucose homeostasis. And by two halves, I mean we need to look at how your body responds to an increase in blood glucose, and then later we'll look at how your body responds to a decrease in blood glucose in order to maintain blood glucose around that set point of roughly 90 milligrams per deciliter. So we'll start here. Here's what we are defining as the set point. Homeostasis is a blood glucose level of about 90 mg per deciliter, and then we'll follow it around. Some stimulus causes blood glucose levels to rise. Maybe you just ate an ice cream cone. And so your blood glucose goes up. There are cells in your pancreas, those beta cells actually, are able to monitor the concentration of glucose in your blood. So those beta cells are the receptor, they are the control center, and they secrete insulin. So they release insulin, insulin travels through your blood, it binds to insulin receptors on cells all over your body. You have a lot of skeletal muscle cells, you have a lot of liver cells. Those cells become, or all cells become very permeable to glucose. Glucose leaves your blood by entering body cells. Your liver cells and your skeletal muscle cells can take that glucose and use it to make ATP, or they can take that glucose and store it as glycogen. Either way, your blood glucose levels go down, and those beta cells in the pancreas are going to detect that. We've returned to homeostasis, and they will shut down insulin secretion. Is this a positive homeostatic mechanism or a negative homeostatic mechanism, feedback mechanism? It's a negative mechanism because the effectors, in this case body cells, including liver cells and skeletal muscle cells, body cells are the effectors. They are what bring about the change in the controlled condition. And these effectors reverse the change in the controlled condition. So remember way back here, our controlled condition was that blood glucose goes up. Our effectors over here cause blood glucose to go down. So this is a negative feedback mechanism. So our next hormone plays a role in the second half of blood glucose homeostasis, and that hormone is glucagon. It is also produced by the pancreas. Different cells produce glucagon. They're part of the pancreatic islets. Glucagon causes blood glucose to increase. How do we get blood glucose to increase? well, you could eat, but glucagon doesn't really play much of a role in causing you to eat. Instead, glucagon causes your liver to perform gluconeogenesis and glycogenolysis, and that resulting glucose is secreted into your blood. And so if you haven't eaten today, that your pancreas is secreting glucagon, and that glucagon is causing your liver cells to release glucose into your blood. And so here's the other half of blood glucose homeostasis, where we'll start right here with the set points, where the set point is a blood glucose concentration of roughly 90, and then, oh, you haven't eaten in a long time, so over the hours, as your cells use that glucose to make ATP, the concentration of glucose in your blood is going to fall. And that gets detected by different cells in your pancreas. They're part of those pancreatic islets, the alpha cells, detect blood glucose. And when it's low, those cells release glucagon into your blood. And the target tissue for glucagon is hepatocytes, your liver cells. And so your liver cells will engage in glycogenolysis. That's the breakdown of glycogen. Your liver cells will also produce new molecules of glucose from non-carb sources. That's gluconeogenesis. And release that glucose into the blood. Your blood glucose levels go back up. And those pancreatic cells detect that, and the mechanism shuts off. So we can put the two halves together. We've been through this whole diagram, only we went through the top half first, and we just went through the bottom half. And this is happening all day long, right? Homeostasis is this constant shifting balance. A little more, a little less, a little higher, a little lower. Let's use this diagram to recap what we can about carbohydrate metabolism. And it will also be a prelude into lipid metabolism and protein metabolism. So we're looking at the body in the fasting state. So we're in the post-absorptive state. There's no nutrients coming in through the GI tract. And blood glucose is starting to fall. That gets detected by cells in the pancreas, and those cells in the pancreas secrete glucagon. We have just recently talked about the effects of glucagon on the liver. Glucagon triggers these two processes in the liver, gluconeogenesis and glycogenolysis, and those are both ways of secreting glucose into the blood. Where we're going to go next involves the rest of this diagram. It's just a little bit of a prelude. So glucagon, which is secreted in the fasting state, can also signal to your fat cells, your adipocytes. Your adipocytes can start lipolysis, the splitting of lipids. So the splitting of triglycerides into glycerol and fatty acids. And this glycerol here, remember, that's a possible substrate for gluconeogenesis. So your liver cells can take that glycerol and make glucose. Another action for glycogen is to trigger skeletal muscle cells to catabolize proteins to form amino acids. Those amino acids can be released from your muscle cells, and your muscle cells are chock full of protein, right? So some of those proteins get chopped up and released into the blood as amino acids, and those amino acids are now a substrate for gluconeogenesis. And so in the next segment, the next lecture segment, we'll talk about lipid metabolism and we'll talk about protein metabolism. I will see you next time.